So, hello and welcome to a webcast on signal analysis. I'm the VP of Proteomics at Say Engineers. Today's webcast will 20 minutes, and I would like to talk about in cell signaling analysis in general, and I would like on tools that are currently used for analyzing cell signaling, including a new and plex assay signaling analysis. This includes phosphoproteoisotopes, but also proteins. And I will show you how you can create your own assay panel. These 1,500 assays. I will complete the webcast study on EGFR signal. Plus, I would leave some time for question and answers. So, Assay Engineers is a Europe-based CRO. We are a CRO and we operate in Germany. We have created a integrated proteomics platform, meaning that we have integrated Olink that's for circulating in biomarkers we use for discovery. And then there's a blind spot in the proteomics side where we use Hyperplex, an assay which is specifically designed for phosphoproteomic analysis, targeted phosphoproteomics. And we have an assay platform called QSM that measures metabolite turnover rate. So we're looking at fluxes, we're looking at reactions that are catalyzed by enzymes, and we give a very comprehensive overview on enzymes that are catalyzing metabolite turnover rates, and we quantify these metabolite turnover rates. Now, when we talk about cell signaling, and this is what I want to focus today, there's a vast number of cell signaling pathways. To date, approximately 1,000 cell signaling pathways have been described, and the number increases by 10 to 15 every year. When we look at cell signaling a bit more specific, you will see there are about 60 major pathways. And cell signaling in general, basically, these networks communicate signals that are detected by the cell membrane, by specific receptors. The information is processed and then communicated to the nucleus, where cell signaling at the end regulates transcriptomics. So you see already at this point that there's quite some complexity in cell signaling, but also there's quite some opportunity because there's a huge number of potential drug targets that you can find in the cell signaling process. Also, by analyzing cell signaling, you can learn about the mode of action of new compounds, and you can also learn how you can combine different therapeutics to develop combination therapies. If we look at the tools that are mainly used today, you see on the left side, you see the Western blot. There's a 10-plex assay, and you see already Western blot is a fairly sensitive platform, but analyzing 10 markers is probably kind of the limit, really, and is kind of a very causal assay, but it's not very holistic. In the middle, you see gene set enrichment, and you see LCMS shotgun proteomics, which is then combined with an enrichment process. And you see here on the left and on the right side, you get very different results depending on which instrument you use, and also in case of gene set enrichment, in the way you use this gene set enrichment process. So depending on the gene sets that you actually use, you can get a very different result. So this is a very holistic platform. Problem is it's not very causal, and it's almost impossible to validate these assays. And on the right side, you see a phosphoproteomics platform, typical mass spec phosphoproteomics. We look at the bunch of phosphosites, and this platform is great if you want to discover new cell signaling pathways. But in the industry, we often really want to make known pathways and well understood and validated pathways available for analysis. And here, phosphoproteomics does not provide very actionable data. The proteomic platform that I would like to introduce you is called Hyperplex. It's a 1500-plex targeted assay which targets cell signaling proteins. So in most of the cases, we're looking at the total protein and we're looking at the phosphorylated forms of this protein. And then we have pre-configured assay panels. You can customize them according to your needs. And here, for example, we look at a panel that we call RTK Ras-Raf-ERK panel, and we have all the major pathways in our platform. So this is a typical experiment with a cell line called A431. It's a cancer cell line which is particularly well suited for cancer signaling. And when we treat these cells with EGF, then the EGF receptor picks up the signal, and the signal is then communicated through the MAPK-ERK signaling pathway plus the PI3K AKT pathway, which then follows to the mTOR pathway. The MAPK-ERK has an impact on cell proliferation and survival, and PI3K AKT has an impact on cell growth and proliferation. And what you also need to keep in mind is that cell signaling has a temporal effect. So there are short-term signaling effects, mid-term and long-term signaling effects. The EGF signaling certainly belongs to the short-term signaling, meaning that it takes a couple of minutes. And it's really important when you prepare your experiment that you pick the right time point in this temporal process to make sure that you look at a relevant sample. Our application scientist will help you designing these experiments, because there's no point in 500 plexing a sample where you missed the right time point. The way Hyperplex works is pretty straightforward. So typically we look at two samples, a control sample and a treated sample. And because for many of these epitopes there are no two antibodies available, so it's not possible to set up this sandwich assay approach, we are combining the sensitivity of a ligand binding assay with the specificity of electrophoresis, meaning that we run an automated electrophoresis step. So we learn and we measure the size of the protein, and the size fractions are then bound to color-coded beads. So at this point, we can make the link between the bead and the color code, and we can make the link to the size of the protein. So this way we can make sure that we look at the band that is exactly running at the exact right size. Then these beads are incubated with up to 1,500 different antibodies, and we read out the information using a Luminex fax machine. And then downstream, we can create heat maps, volcano plots, and we can visualize signaling pathways like I've just shown for the Ras-Raf-ERK signaling pathway. We have validated the platform on different levels. So first we cross-compared to a traditional Western blot. We also compared to mass spectrometry. Then we have verified the information against the specifications that we knew about the size of the protein and also the specificity of the protein. We used an alkaline phosphatase step to dephosphorylate the phosphorated proteins, and then you can make up the mass balance. And the fifth layer simply is conclusiveness, because if on a specific pathway which is upregulated, maybe you put 10 assays on this pathway and 9 go up, and maybe one assay doesn't show any changes, then you know here's an issue. So this way you can use, in signaling networks, you can use conclusiveness as a tool to validate these assays. So now to configure the assay panel according to your needs, please go to our website assayengineers.com/configurator, and you'll find this Excel spreadsheet that gives you an overview on the 1500 assays that we have up and running, including the antigen name, including the phosphosite. There's information on the gene names and also information on the size of the protein. Then you go to the second macro, you select the species, and then you go to the third macro, and here the information on cell signaling pathways is structured in two different ways. First, you can look at the cell signaling pathways, and you can also look at disease-centric signaling. So let's start with the cell signaling pathway macro. Here, for example, there's information on the MAPPK signaling, there's information on RAS signaling, and there's pre-configured assays for the PI3K-AKT signaling pathway. Here, for example, we have 373 markers for the PI3K-AKT signaling and 464 for the MAPPK signaling. There's also information on developmental pathways like hippo, hedgehog, and notch signaling pathways, TGF, Wnt signaling. There's also information on stress and metabolism. For example, if you're interested in the P38 MAPPK signaling, there's another 67 antibodies that we have up and running for this signaling pathways. There's information on immune signaling assays. There are panels pre-configured for cell death and also G protein, and then there's some other signaling processes including caterin, FOXO signaling, and integrin signaling. You simply check the boxes that you're interested in, and then there's a second layer, the disease-centric signaling, and as you can see, we are pretty heavy on the cancer side. So within cancer, there's this kind of hallmark panel that we have pre-configured for you, but also specific and disease-centric panels. We have panels on cell cycle and proliferation in general, and then there's also on CNS disease and infectious diseases. We have disease-centric assay panels up and running. You can cover immune processes, T-cell maturation, and again some other steps including platelet and cell migration. A third layer is pre-configured panels that target mTOR, PI3K-AKT signaling or RTK, MAPPK signaling pathways. And there's also information on druggable targets available. Simply tick the boxes here and then push select antibodies. And then here in column F, you will see all the antibodies that we suggest. Now, this list is not set in stone. You can remove markers that you're not interested in, or you can add additional antibodies that you wish us to use, and you just send them along with a sample, and we will run them for you. Right. So with these assays, you can now run all sorts of tissues. We work with cell lysates, organoids, cell lines. You can analyze xenografts, tissue sections, fast frozen sections, as well as FFPE sections. But I should mention that for FFPE, the choice, the selection of antibodies is a little limited, and obviously also with PBMCs or sorted cells. Typically, we need 20 to 60 microgram of protein, but this depends also on the number of assays that you want to run per sample. So overall, Hyperplex is a very comprehensive platform. It's the most comprehensive signaling platform on the market today. We will provide you with custom data analysis reports, including heat map maps, volcano plots, and also pathway maps, and to make sure that stakeholders really get a very deep understanding of the signaling processes going on. Our biomarker panel comprises expert-created biomarkers, including cell signaling networks and disease panels. Also, we're not trying to lock you into our platform. Most of the antibodies that we use are from cell signaling technologies, and when you're interested in specific reagents, we will make the ordering number of these reagents to you available. So in case you want to carry on working with some of these markers, just let us know. So if you want to learn more about the platform, please contact us at hello@assayengineers.com, or you go to calendly.com/proteome/cell signaling analysis, and you schedule a meeting with an application scientist. And if you want to download the assay configurator, please go to assayengineers.com/configurator. Now, with this, I would like to conclude my talk, and there's time for another one or two questions. So I go to the chat, and the first question is about turnaround times and cost. So for a typical project, turnaround time is between 5 and 6 weeks from the day samples arrive in our lab. And the cost depend a little bit on the number of samples and the number of markers. So typically cost per data point, meaning one sample, one marker, is somewhere between $9 to $20, depending on throughput. All right, I hope this answers the question. There's a second question on the assay configurator. Yeah, so this is a pretty useful tool. So even if you don't want to work with our assay platform, this assay configurator is a nice tool to get an idea about the markers that are available. And you can download the assay configurator on our website, assayengineers.com/configurator, or you send us an email at hello@assayengineers.com. Thank you very much for your time, and please let us know if you have any questions and we can help you. Have a nice day.